Okay, well, I like that.
Okay. Thank you, everybody, for joining us once again for our Thursday night seminar series here at Stone Lab. I'm sure you're all wide awake after a very active and fun-filled week out here on Lake Erie. Thank you to those of you joining online. Please feel free to type in any questions for the speaker at the end, and we will be sure to announce those questions from you to the crowd. Without further ado, it is my pleasure to introduce to you Sarah Thomas, who comes to us from Michigan DNR, just across the way here in Southeast Michigan. A fisheries biologist who has some connection to Stone Lab. She was once also a student here at Stone Laboratory and is now a bigwig in Michigan state agency institution. So I will let you talk about her. I will let her talk about herself and her work there at Michigan DNR. So thank you, Sarah, for being here and for bringing your lovely family, your husband and 2 daughters here who are joining us. So with that, I'll turn it over to you.
Excellent. Thank you, everybody, for showing up on this beautiful Thursday evening. So a little bit about myself. I was born and raised in Northwest Ohio. So I am a Buckeye. I don't admit that too much up in Michigan anymore, but I got a Bachelor of Science in Water Resources and Environmental Biology from Heidelberg University in Tiffin, Ohio. And I went there specifically because of their water resources major in the water quality lab. And I was fortunate enough to work all 4 years at school in the water quality lab. And that's what I thought I was going to do. My dad was a farmer. I thought I wanted to look at non-point source pollution and save the world by cleaning up the water. And then I took fish ecology here at Stone Lab, and it completely changed my career directory. And I decided that fish in the Great Lakes was where it was at, and that was what I wanted to do for my career. So I then the following summer interned with the USGS at Sleeping Bear Dunes National Lakeshore up in Traverse City, Michigan. And I worked on doing water quality sampling on the inland lakes in the park, and Lake Michigan was right there. And I was like, oh, I really want to work on that lake and not these teeny tiny lakes. So I went to the University of Michigan and got my master's in aquatic ecology and did primarily a modeling and GIS study looking at the potential habitat above dams and how that could increase the population of podonamadrous fish, such as salmon and walleye and sturgeon, if those dams were removed. So I got a really great analytical baseline. I didn't have a lot of field work myself, so I was always the first one to volunteer when other graduate students needed help. But that led me to my dream job, working as a fisheries research biologist on Lake Michigan for the Illinois Natural History Survey. And I did that for 14 years. Basically spent May through October out in Lake Michigan doing primarily nearshore work, looking at habitat and invasive species, native fish interactions. I did a lot of diet studies. I got pretty heavily into zooplankton towards the end, and I will talk about that at the beginning of my talk. And then found myself in 2016 moving to the state of Michigan to accept a job as the Lake Erie Unit Manager for the Michigan Department of Natural Resources. So I am the supervisor for 2 biologists, a technician supervisor, and then we have 2 technicians at my office as well. We also have a large staff of AIS folks who are working on grass carp and red swamp crayfish response. So little tidbit, if anybody knows, Michigan DNR biologists cover more freshwater acres than any other state in the country. It's kind of sad that I cover 12.5 counties and have 2 biologists for that entire area. And then also as the Lake Erie Unit Manager, I'm involved in the management of the St. Clair Detroit River System and Michigan waters of Lake Erie as well. I also became more heavily involved with the American Fisheries Society. After moving to Michigan, I served as the Michigan chapter president, and I am finishing out my role as the president of the North Central division this year. So if you're interested in fisheries, I do encourage you to look at becoming an AFS member as well. Really great networking opportunity and student opportunities for that. So what I'd like to talk to you tonight may seem like, what in the world, Sarah? These topics have nothing to do with each other, but I wanted to talk a little bit about the various stages or scales and how everything is important when you're talking about natural resources, from the littlest thing to the big thing. So the micro scale, if we're talking about, you know, this, right? Most people don't think about algae, right? Until it's looking like this and boom, it's in our face. And then we're like, oh, this is a problem. Except when you don't have long-term data sets looking at what happened before it was a problem and now it is a problem. So it's always important. There's a lot of data gaps when you're talking about the really small organisms. They're underappreciated, generally underfunded, but really important, right? Because those small organisms are the key to everything. They're the base of the food chain and the trophic chain. So even though they're challenging to sample and take a ton of time to process, usually in the lab, really, really important that we don't forget about these microscale organisms. The meso, what I'm kind of going to refer to as those fish that everybody sort of thinks about, right? Bluegill that people are going fishing for, your yellow perch, your walleye. They're what brings everybody together, right? They're what gets people out in the water and caring about fishing. But because they're so utilized, it's really important that we have a good background understanding of them. And so really we need to utilize the expertise and resources and have a lot of partners looking at these organisms. And then finally, our mega organisms, such as the muskellunge. They're big, right? They're going to attract a lot of public attention. People will spend a lot of money to have the opportunity to go out and catch these types of fish. And when we have these big charismatic species, they're a really good opportunity to use them for outreach and increase support for other management and restoration activities. So this is kind of where we can grab the public's attention with something big and wow, right? So the first thing I'd like to talk about is some work that I did when I was in Illinois and working on Lake Michigan. And so what we did was look at microzooplankton. And so for the purposes of this, microzooplankton were what we considered rotifers, zebra mussel or quagga mussel veligers, and then copepod nauplii. So these are the really small members of the zooplankton community, not the slightly bigger members, the crustacean zooplankton, like the Daphnia that most of you may know. So microzooplankton are a really important part of the zooplankton community. They're very good at nutrient recycling. They have high growth rates, quick population turnover, but they're often overlooked because they're so small. And you really need specific small mesh sizes to accurately assess them. And the problem is that the majority of Great Lakes studies are not using those methods to quantify the microzooplankton well. So since 1973, only 4 out of 25 studies used mesh sizes smaller than 35 micrometers, and that's really what you need. You need something really small to really quantify these small organisms. And so one of the reasons we really wanted to look at this in Lake Michigan is that these microzooplankton are a major component of Asian carp diets in the Illinois and Mississippi rivers, and we wanted to know what that prey base was in the instance if, heaven forbid, the big-headed and silver carp were to get into Lake Michigan. So our objective of the study was to compare the effectiveness of two different sample methodologies to collect microzooplankton in both harbors and open waters of Lake Michigan. And we wanted to do this to provide more accurate estimates of available microzooplankton prey to the Asian carp and other planktivores. So here are some of my super cool rotifer species. So the first method we did was our traditional macro zooplankton method, and this is what we had done at the station since 1988. It's what most Great Lakes studies do, use approximately a 63 mesh with a 1/2 meter diameter plankton net, which was pulled up from a 1/2 meter above the bottom. And then our new method, specifically to target the microzooplankton, used a 2.5 liter Van Dorn sample that we lowered each meter in the water column, and we collected water from each meter down. That was integrated into a bucket and then filtered through a 20 micron mesh pipe sieve. And then we counted the rotifers and Nauplii and veligers using identical laboratory methods. The first sampling that we did occurred monthly, and that was May through October. Again, we did open water and harbor over here, and then we also sampled the drowned river mouths over on the Michigan side. We ended up with a total of 43 locations. So using both methods at both locations, and all of our water depths that we sampled were less than 10 meters in depth. So looking at the results, when we take a look at the density, so the number of organisms per liter, when we used our traditional giant zooplankton nets, we found about 40 rotifers per liter. But when we used our specialized method, we found an order of magnitude higher of rotifers, and we found 580 per liter. The difference wasn't quite as drastic with the veligers. It was about 6 times higher for veligers using the specialized method, and about 3 times higher, 4 times higher for the Nauplii. And then if you take a look at the biomass, it was the same thing. When we used the specialized method, obviously we found a lot higher biomass using that smaller mesh size technique. If we take a look at the individual rotifer genera, the species that had the biggest difference between the methods was Polyarthra. These were one of my favorite. They look like tiny flies underneath the microscope, and they're one of the smallest rotifers. So not really that surprising that we saw quite a difference between the 63 and the 20 mesh, but basically over 300 times higher using our specialized method to target these smaller organisms. A couple of the other more common ones, Caratella, we had about a 20 or 15 to 75 difference, same with the Synchaeta. Trichocerca was a little bit less. Tylotricha was really interesting. I think we only detected it in one sample with the 63 micron mesh net sample, but it was one of our most prominent species in the 20 micron, and then Theosoma too, the same difference. So definitely, you know, it wasn't as dramatic with some species as it was for others, but we caught more with the smaller method to target these species for every species. So then we wanted to take a look to see if we could use this data to take our old data and try to get a better estimate of what our microzooplankton densities would have been with the 63 micron. So we ran regressions looking at the 60 relationship between the rotifer density and the 63 micron mesh and the 20 micron mesh. And for all rotifers, it actually was a pretty tight, good relationship with an R squared of 60. So obviously, you know, we caught more with the 20 micron. So we felt like we could pretty confidently back predict, and correct our old estimates if we wanted to look at all rotifers together. But when we looked at individual species, it definitely wasn't as tight of a relationship. But 30% in ecology is not terrible. So definitely still showing that relationship with catching more in the 20 micron compared to the 30. And then, so what does this all mean as far as looking at the community composition? So with the 16 micron mesh, rotifers accounted for about 50% of the density, and your typical, what's most studied, cladocerans and copepods accounted for a lot less. But when we looked at our method, combining the 63 and the 20 micron mesh, we saw that there was even, that bigger difference, that rotifers are a lot bigger part of the community than what most studies are sampling, and that the traditional cladocerans are a really, really tiny portion of the zooplankton community if you're looking and using methods that are accounting for those smaller critters. And then this has even more of an impact when we look at the biomass. So rotifers were reported as accounting for only 5 to 20% of zooplankton biomass in the Great Lakes. But remember, these past estimates were used on the macro zooplankton sampling methods. So people like we were at the station using 63 micron or more. And a lot of the work that's done in the offshore, they actually use a 153 micron mesh net. So they are definitely missing a lot of these critters. So looking at our sampling using the 63 micron, it looks like cladocerans were accounting for most of the biomass here in the blue, almost 50%, copepods about 20%, and then the microzooplankton were only accounting for 30% of the biomass. But when we looked at combining the cladocerans and copepods that we got in the 63 micron mesh net with the microzooplankton that we got in the 20 micron sampling, what we see is that the biomass is actually a way higher contribution of those small microzooplankton. So rotifers accounted for 51% of the biomass, even though they're teeny, teeny, tiny. There's so many of them that they are actually making up half of the biomass of the zooplankton community. And then the cladocerans and copepods are a lot less important when you're looking and including sampling methods that get these really small critters. And so when we looked at some other data, here were our results from the 20 micron, and we showed about 400 per liter in Lake Michigan, higher in the drowned river mouths and the harbors. And then these were results that used the macro 63 micron, and you can see it's a lot lower numbers in Lake Michigan and Lake Ontario in these various time periods. But when the EPA sampled the Missouri River and Mississippi River and Ohio River using a 20 micron screen, they got very similar numbers to what we were seeing. So definitely, if you want to look at these small members of the zooplankton community, you have to use the more specialized method that's specifically targeted to these critters. So what does this all mean? So basically, traditional sampling approach significantly underestimates the microzooplankton abundance and its relative importance. And this can have big impacts on fish diets and what fish are eating. The methodology also impacts the community composition data. So this can be important if we have a new invasive coming in and having an understanding of what species they're impacting the most. For example, our 20 micron samples were dominated by Polyarthra, those small little critters that look like the flies, while other studies using the bigger mesh zooplankton nets were dominated by Conocylus. And that's a colonial rotifer, so they all stick together, so it's a lot bigger, so very easily captured by those bigger mesh sizes. And then Chelicotia has these super long spines, so even though the body itself is small, the long spines make it more easy to get captured by those larger mesh sizes. So to get an idea of the community composition and what fish are eating and what might be impacted by invasive species, we really got to know and use the smaller mesh size method. So without this information and using the targeted methods, modeling efforts on the food web are less informative without this accurate microzooplankton data. So for instance, the prey resources available to the bighead and silver carp in the Great Lakes were underestimated in a study that was done in 2010. The zooplankton biomass that they used did not include rotifers, and our data showed that the biomass could have been up to 3 times higher. And we showed that 2 of our sampling sites had total zooplankton biomass that could have met the energy resting requirements for these invasive species. And so we did a follow-up study, and we went to even more sites, and we brought in partners from Michigan who were able to sample more of the drowned river mouth lakes for us over on the Michigan side. And we brought in folks from Wisconsin who were able to sample Green Bay for us. And so we sampled all of these sites using the dual methods and looking at the microzooplankton, and ran an energetic model. And so what we found was that these sites with the green actually had enough zooplankton biomass to have positive growth in silver carp. And before, there hadn't been a study that had showed this before. So variation in the near shore zooplankton community can have impacts on growth of both native and invasive species. So the good news is that there is still likely not enough plankton biomass to support growth of bighead carp in the open waters of Lake Michigan. But the small areas of several of those drowned river mouths in Michigan and Green Bay do sometimes have enough zooplankton biomass to have growth of these species if they're able to make it. So the take home here is that relatively small areas and connected waterways and the teeny tiny species that live in them may play an important role in the establishment of filter feeding invasive species. But this isn't all gloom and doom, because this gives us really great insight into areas that we really should continue to monitor and keep a close eye on. And that if these species do make it past the barriers that they have in the Chicago Sanitary and Shipping Canal, that these would be really good areas to target control actions as well, because this is where they're going to be if they're going to want to grow. So that was my spiel on why the really smallest parts of the aquatic community are important to understand and make sure that we are sampling them with the proper methods. Next, I want to talk about those middle species are most common that people think about, and why partnerships are so important when we are looking at these species. And so I'm going to take a look at a collaborative study that we have done on Lake St. Clair with Ontario Fish and Wildlife and US Fish and Wildlife as well. So a little bit about Lake St. Clair, if you're not too familiar. The heart-shaped lake just to the north of us here, it is about 430 square miles. A third of it is in Michigan waters, 2/3 is in Ontario waters. It has a very diverse fishery. I don't want to knock Lake Erie, but it is definitely one of the top fishing destinations in the US, multi-million-dollar fishery. Tons of recreational boating. I've heard the stat that Lake St. Clair has a higher, the second highest boating density in the US next to Florida. There's tons of canals, just boats everywhere on this lake. Commercial shipping, so everything that's going down here has to go through Lake St. Clair. Very complex system, just like Lake Erie, lots of number of variables, invasive species, and always changing. As I mentioned, it has a huge fishery. Lake Erie and Lake St. Clair combined make up less than 1% of the total surface area of Michigan's Great Lakes waters, but we lead in catch rate and angling intensity. So Erie and Lake St. Clair combined had about 19% of the total effort that anglers spent on Michigan waters of Lake Erie, but those two lakes having only 19% of the effort account for 40% of the catch. So when people go on Erie and Lake St. Clair, they are catching fish, and they are catching a lot of it. So the catch per angler hour for Erie is almost 2. Lake St. Clair, it's a little bit of 1.5. And then Huron, a little below 1, and Michigan Superior, less than 0.5. Angling intensity, we quantify by the number of hours per square mile of the lake. So Erie is off the charts at 3,000, Lake St. Clair a little bit above 2,000. And then you can see in comparison, how low that is for Huron, Michigan, and Superior. So really important fishery, brings in a ton of money to the economy in this area. So it's one of the reasons that we tried to study it, but in the past we didn't do a great job, especially considering that it's jointly managed between Michigan and Ontario. So in the past, we had some volunteer angler diary programs between the two countries. There was some coordinated creel, which happened about every 10 to 15 years. We cooperated on a muskie acoustic telemetry study, but for fishery independent studies, like surveys, there wasn't really a lot done together and that was coordinated. So currently in Michigan, our primary work on Lake St. Clair is spring trap nets. You can see in Ontario, they did fall trap nets, so not really very good comparable data. Michigan, we do spring and fall bottom trawls and then fall electrofishing. And then Ontario also beach seined. So there was nothing that we did the same where we compare and say, hey, what trends are you seeing? Because we were sampling at different time periods and using different gears. And so we didn't have good data to look at. So we wanted to try to get something together. So in 2018, the Ontario Ministry of Natural Resources and Michigan DNR met to discuss, we need to fix this and we need to do a better job of coordinating our surveys and getting complementary data. And then the U.S. Fish and Wildlife Service Early Detection Monitoring Program, which is primarily focused on looking at invasive species, joined on, and we decided on a 5-year rotating sample method that would include nearshore small fish fyke netting, fish community gill netting, connecting channel assessment, so looking at the St. Clair River and the Detroit River, and then a nearshore fall electrofishing study, which would look at all species, but also have a focus on muskellunge. So COVID threw a wrench in a few things and we couldn't start this until 2021. But the first thing that we did was our fyke netting, and we did a total of 110 sites. Michigan DNR did the ones in the red, U.S. Fish and Wildlife Service did the ones in the green, and Ontario did the ones in the blue. And out of those 110 sites, we caught a total of 27,000 fish for an average of 246 per net. We identified 45 species, so that's pretty awesome. The most common was bluegill, followed by largemouth bass, smallmouth bass, and gizzard chap. These were our catch rates, so you probably noticed something that pops out right away. Bigger bubbles mean we caught more fish, so the bigger bubbles are all on the Canadian side. If we break the fyke net catch down by agency, Michigan found 23 species in our fyke nets for an average of 44 fish per net. Our most common fish were bluegill, smallmouth bass, and largemouth bass. U.S. Fish and Wildlife Service found a few more species and they had a few more fish per net. Smallmouth bass and bluegill were also some of their top species. Largemouth bass was a little bit lower. And then we had Ontario, who caught 42 species, so a lot more species than we did on the U.S. side, and 566 fish per net. So way more fish, as that bubble graph was showing, but similar fish for most common. But bluegill, you can see here, was 196. Bluegill here over here in Michigan, 10 per net. So definitely a pretty big difference. We did a species richness hotspot analysis. So cold spots, fewer species. In the blue, red was the highest species richness. So our hotspots for species richness, definitely all here on the Canadian side. So, any difference in habitat? What do you guys think? Yes, definitely. So here we have a fyke net, some beautiful natural shoreline, and here we have a fyke net and seawall. So the U.S. side of Lake St. Clair is 90% hardened shoreline, primarily seawall. There's some rock riprap. The Canadian side is only about half hardened, and they have almost 30% still marsh wetland remaining. So yeah, you can see seawalls more than double in the Michigan side compared to the Ontario side. So if we take a look and break down where our fake nets were set and what the shoreline type is, the number of fish was highest along sand beaches and rocky shorelines, lowest along seawalls and vegetated banks. If we take a look at the number of species or the species richness, sand beaches were the highest and seawalls were the lowest. So definitely an impact of habitat. There are a little bit stricter regulations to what you can do to your shoreline in Canada. And so there's a really big push, particularly from Fisheries Division, to move towards more bioengineered shoreline techniques and trying to move away from seawalls, because we can see what the impact is on the fish community when you have this large amount of hardened shoreline. So the next year we went out and we set gill nets. And these were set in late June, early July. They were about 53 meters long and had 8 mesh sizes in them. We did a total of 75 sets, and we classified those into shallow sets, which were in water less than 3 meters deep, and deep sets, which were in water greater than 3. So this, again, just showing you where all those were. Overall, we caught 2,000 fish in the gill nets. 33 species were identified. Our most common were yellow perch, shorthead red horse, white perch, gar, walleye, and smallmouth bass. And so with the gill net catch, there wasn't really a big difference. There were similar numbers of fish caught between the US waters and the Canadian waters, but what we did see was that there was a slight difference in catch between the shallow nets and the deep nets. So we caught a few more species in the shallow nets, 28 versus 23. A few more fish, but really not a statistical difference. And the most common fish in the shallow was gizzard shad, and yellow perch was the most common fish in our deeper sets. And then in 2023, we did the fall electrofishing survey. And this was done in late October, November, so water temperatures were pretty cool. We electrofished for 30 minutes per site for a total of 37 sites. And we did try to select the available habitats for this. So we tried to sample sites that were along seawalls, that were along natural shorelines, that were rocky, that were vegetated. And this was the gear that we caught the most fish in. We caught a total of 45,000 plus fish, and we identified the most species, 47, using the electrofishing. We didn't catch a ton of young of the year muskie, but we did catch 15. And the most common fish were gizzard shad, emerald shiner, and brook silverside. So more small prey fish species rather than the larger sport fish. Again, we did see a difference between those fish caught on the Michigan side and the Ontario side. So Michigan was 110 fish per 10 minutes compared to 677 in Ontario. The number of species was pretty similar, though. They just got 3 more in Ontario. And so the primary driver between the high numbers in Ontario was that they caught a ton of gizzard shad and emerald shiners. So lots of those small prey fish species. But when we take a look at yellow perch, they actually caught fewer than we did in Michigan waters. We did detect multiple at-risk species in the sampling. So eastern sand darter, lake sturgeon, mooneye, northern madtom, pugnose shiner, river red horse, silver chub, and spotted sucker. And this is really cool because on a lot of these species, we don't have a lot of great data. So this will be a really good long-term survey that we will have with this data so that we can track the trends through these at-risk species through time. And we didn't have this information available to us previously. So the take home of this is that there are huge benefits to partnerships. We would not have been able to sample a lake as large as Lake St. Clair this intensively without the partnership with the U.S. Fish and Wildlife Service and Ontario. It's been beneficial to all of the agencies involved, monitoring and having that long-term trend daily available once we continue this 5-year rotation and having a good idea of what's happening through time, monitoring for newly introduced species. This is complementary data to our other research and surveys that are going on. We can look at our creel survey data and see how trends and what the anglers are catching is comparing to what we're getting in the gill nets and things like that. And again, it really highlighted the need and importance for nearshore habitat restoration, and that seawalls aren't going to cut it. We need something different so that we get higher species richness and diversity and more numbers. And then finally, I want to conclude with one of my favorite mega species in the world, the lake sturgeon. And so we have closely monitored lake sturgeon in the St. Clair system for 30 years now with Michigan Department of Natural Resources. And I was laughing because Brian was telling me about how old your research boat is. And our lovely Channel Cat here is even older. Channel Cat was built in 1967 and is definitely showing its age. But for now, it's chugging along. So prior to 1996, we knew sturgeon were in the St. Clair Detroit River system, but we didn't really have any good data. There was a small recreational fishery, but it wasn't monitored. There were no targeted assessments. And they worked on a Michigan statewide rehabilitation plan. And they said, hey, there are a lot of information gaps in your system. So starting in 1996, Michigan DNR started our lake sturgeon study to annually document the status and dynamics of lake sturgeon in the system to help with the conservation and management of this threatened species. And since 2006, other agencies have done a lot of great work as well. The U.S. Fish and Wildlife Service, Ontario, and USGS have done studies on the Detroit River, the Upper St. Clair River, and Lake Huron. And so together, all of this information has given us a really great data set on sturgeon in the St. Clair Detroit River system. We've used various gear types through the years, but the two predominant survey gears that we have used since the very beginning and what we are using today are set lines and bottom trawling. So the set lines, there's these long lines with baited hooks that and then the bottom trawl is used in Lake St. Clair. We use Goby as the bait for the set lines. That's what they found to work the best, and it's free, so hey. When we collect the lake sturgeon, we get a lot of data from them. So we take their length, weight, and girth. We identify their sex and maturity if possible. On smaller fish, we take a fin ray for aging. These are such long-lived fish that if the fish is too big, it's just too hard and you can't get an accurate age estimate on the fin ray. We note the number of lamprey scars and how deep they are. We look for any abnormalities of the fish. And if it doesn't already have a tag, it gets a pit tag. And on really large fish, we put the metal tag with a number on it as well. So I want to talk a little bit about the sturgeon spawning areas. So unfortunately, a lot of the historical spawning areas in this system were destroyed when they put in the shipping channels and other human actions. One of the last remaining natural spawning areas is right here at the Blue Water Bridge where Lake Huron enters Lake St. Clair. This is one of the fastest flowing sections of river in the United States, but the sturgeon love to spawn there. So the X's are historical spawning areas. And then starting in 2004, I believe, we started to construct some artificial spawning reefs to put some of that rocky habitat back in the rivers that had been taken out so that the sturgeon had more areas to spawn on. So the triangles are the new reefs that we have constructed. So this is sort of what that looks like. They were built from barges. We had to very carefully select these areas where they would not interfere with shipping traffic and that they had the right flow characteristics. So one of the first ones that they built was filled in with sand within a few years. So then they did a lot more geotechnical analyses prior to putting these reefs in to make sure that that wouldn't happen again. But within a year, we're already seeing sturgeon using these newly constructed spawning reefs. So that was really cool. Studies have confirmed lake sturgeon spawning, and we have collected viable eggs and larvae at 5 of these reef sites. We've also seen evidence of 16 other native species using these reefs for spawning. And one of the really cool things is that we documented the first whitefish spawning in decades in the Detroit River on one of these artificial reefs. And an additional 13 native species have been using the reefs just for hanging out and feeding and other activities. So from 1996 to 2023, we have caught and released 3,713 lake sturgeon. The length range has been from 9.6 to 75 inches long, so teeny tiny to really, really big. And we have recaptured 769 unique sturgeon, and they have been recaptured 1,364 times. So I think we have one fish who loves us and has been recaptured 8 times. So pretty cool. We have a 21% recap rate. Taking a look at our set line size distribution through the years. So this orange color is 2016 to 2019, and the blue is 2021 to 2025. So what I want you to notice is that when we take a look at the smaller fish in the more recent years, we are catching more smaller fish. So this is really good, and we think that those spawning reefs might be contributing to that, and that we're catching more smaller fish because we're getting more reproduction in the system. We have an awesome, healthy population in the Scudder's system. So these are the year classes that we have found through our study from 1996 to 2004. And you can see we have sturgeon from every year. So that's really awesome. It's not something that every system with lake sturgeon can say. So we are really excited about that we have healthy reproducing fish and that we're getting recruitment every year. So when we take a look at all of these gill nets and set lines and things that we have done through the years, our set line catch has been pretty steady. The trawl catch has declined, but we don't think that's related to numbers of sturgeon declining. We have seen a drastic improvement in Secchi disc reading in Lake St. Clair, and the water is much clearer than it used to be. And so we have a very good relationship between Secchi depth. As the Secchi depth increases, our sturgeon catch is declining. And then also with the increased Secchi depth, we're getting more vegetation growth, which also makes it a lot harder to trawl. So we think the declining catches in the trawl are more in relation to environmental variables rather than the sturgeon population itself. But our set line catch is steady, which is awesome. We've shown that the pit tag retention is nearly 100%. So that's great for our modeling. We get our recap numbers. We can have pretty good confidence in our population estimates. The mono tag retention is not quite that high, but on older fish, it's definitely up there. As I mentioned, more juveniles are being caught on the set lines recently. It's a really good sign that we're getting more young fish into the population. And when we do the bottom trawls in Lake St. Clair, we are predominantly catching really big adults. Another really unique aspect of the sturgeon population in the St. Clair Detroit River system is that we do have a recreational fishery. So back in the day, lots of people caught sturgeon. Some people threw them on the banks and thought they were trash fish. Some people were catching them from caviar, but there weren't a lot of regulations, and that's one of the reasons why lake sturgeon were threatened, in addition to all of the habitat destruction that humans did. But in 1928, there was a statewide lake sturgeon harvest ban. Starting in the 1950s, some harvest was allowed in Lake St. Clair. For the next 45 years, there were various changes, but they all made it more restrictive for regulations on lake sturgeon in the system. And then starting in 1999, lake sturgeon angling in Lake St. Clair and the St. Clair River only, harvest was allowed July 16th through September 30th, and your fish had to be between 107 and 127 centimeters. I believe that's 42 to 50 inches in order to keep the fish. If you harvest a sturgeon, it must be registered. So you can do that either online or through a toll-free phone number. Starting in 2008, a catch and release season was added, and that included the Detroit River as well. So still no harvest is allowed in the Detroit River, but catch and release is. And that catch and release season was tacked on to the end of the harvest season, and that ran through November 30th. And then I'll talk about this in a little bit more detail later, but in 2018, we made the catch and release season even longer, and that went through the winter until March 15th. So having a lake sturgeon fishery is pretty unique. There are only 4 populations in Michigan where harvest is allowed, and there's only 7 populations where fishing for lake sturgeon is allowed. And Lake St. Clair River is definitely the biggest one. So we do have several ways that we monitor the recreational fishery. First is the Angler Diary program, which gives us information on the angler catch rate. We have our harvest database, which gives us information about the angler demographics, the harvest rate. We do have a master angler database, and if you catch a lake sturgeon that meets a certain length threshold, might be 60, 65. I'd have to check on that. You can get a master angler patch, the recapture database. So when anglers catch a fish that has that tag, if they call us and let us know where they caught it, what the tag number is, and then we provide them information and let them know when we originally caught that fish, how big it was when we caught it. And when they report a tagged fish, they get one of these sturgeon cooperator patches. So that's kind of cool. Some people have a whole collection. And then our tagging database gives us information on the exploitation rate and an approximate survival estimate. And so one of my colleagues, Andrew, published some information about the recreational fishery in the 2020 North American Journal of Fisheries Management. And so a lot of this information is from that. So this is our size distribution of sturgeon that we have caught in our survey since 1996. So here is the fish that are in that harvest slot limit. So the slot limit is protecting about 80% of the lake sturgeon that are out there. And primarily, these really big fish are going to be your females. So we are protecting a lot of those spawning size females. So we are definitely seeing an increase in interest in lake sturgeon fishing in the system. So if we take a look at here, we have number of fish per 100 sturgeon tagged in the year, and anglers are catching way more fish recently. So people are loving it. They have sturgeon fishing tournaments. They take a picture, fish gets thrown back in the water right away. And our harvest rate is increasing as well. So this is the number of lake sturgeon harvested in the St. Clair River and Lake St. Clair, number of fish. So what you will see is that we seem to have maxed out at about a total of 14 fish harvested per year. We know there's probably some poaching going on, but this is a really passionate community. And so if these people see somebody poaching, they are on the phone with our conservation officers right away, and they are not letting people get away with it if they are seen doing that. So, so there is some harvest, but really low considering the population density in this area and how many people are fishing for them. So taking together all of the survey data and using Michigan DNR data and U.S. Fish and Wildlife data, Chayot 2023 ran a population estimate. And so we are guessing there are about 20,000 lake sturgeon in the lower Huron, upper St. Clair River portion, about 6,500 sturgeon in the lower St. Clair River, and about 6,400 in the Detroit River system. So what that gives us is a total system population of about 33,000 lake sturgeon. So definitely way lower than what we saw in the 1800s and 1900s, but a really great success story for a species that's in much lower numbers than other systems. And so we are trying to bump these numbers up even more. So many of you may know that they are now raising lake sturgeon and releasing them into the Maumee River here in Ohio, and they do a big outreach event at the Toledo Zoo with those. They are introducing some into the Flint River up in Lake Huron. So trying to get those sturgeon numbers back even higher, closer to what they were before humans came along and messed everything up. So a summary of the recreational fishery, as I mentioned, really popular, very unique opportunity to be able to catch one of these awesome fish. So through 2018, the sturgeon fishery drew anglers from 7 US states, 1 Canadian province, and 37 of the counties in Michigan. The mean exploitation rate is approximately 0.16% of our total population. And in the Michigan sturgeon management plan, we want the harvest exploitation to be below 2% to maintain a stable population. So well below that, those 14 fish per year are not having an impact on the population. We've also shown that the calculated survival of lake sturgeon has not changed over time. So the recreational angling, even though it's growing in popularity, is not affecting population mortality. These are tough fish. As I mentioned, we've recaptured that one guy 8 times. They're really good at surviving catch and release. And because we had all of this great data and a good understanding of the sturgeon fishery, when the anglers came to us and said, hey, we love sturgeon fishery and we really want a longer time frame to be able to catch these fish. We had the data to say, yes, you guys are not negatively impacting the population. We can extend the catch and release season with confidence and give you guys these extra months to have catch and release opportunity for these fish. So that's why it's important to have these long-term data sets and really have good combination of the survey data and looking at the angling data as well. Again, I wanted to highlight that these mega fauna charismatic fish really attract a ton of public attention, and we can leverage that towards getting people excited about the resource and wanting to do more and learn more and protect other things. So many of you may have seen this. This is my U.S. Fish and Wildlife Service colleagues who caught this 6-foot, 10-inch, 240-pound sturgeon in the Detroit River several years ago. They posted this on their social media and it went absolutely crazy. It went viral. Some of the comments will really make you think that our science education needs a lot of work, but this is one of our Michigan U.S. Congress women. So, lots of attention, drawing in legislatures and things like that. We have an entire festival dedicated to sturgeon in Port Huron. Big attention here with the Toledo Zoo release and highlighting habitat in the Detroit River. So to wrap up all these seemingly disparate pieces of research and management, the main take home I want all you young students to have is that all aspects of aquatic ecosystems are important to consider, no matter how small, from the tiniest organisms to the big organisms, they all matter. They all should be studied. And that the combination of fishery dependent and independent data, so the surveys that we do, and then also looking at the information that we collect from the angling community is key for informed management decisions and making sure that we have regulations that are protecting our fish populations. Partnerships are invaluable. One agency can't do it all alone, particularly when we're working on big systems such as the Great Lakes. So really making sure that we are working together and leveraging our resources and money and funding, and then leveraging that information and charismatic megafauna into actions for habitat and other species, and giving young kids the opportunity to have hands-on science activities and where they're going to be able to see animals and critters and get out into the wild and really have an experience where they're going to want to protect our natural resources, contact their legislatures when they're older. So these are my kids who are in the back, who have gotten to tag along with mom to work. several times. So I encourage you when you and if get to that point, if you have cousins, nieces, nephews, and whatnot, anytime you have the opportunity to get them out into the field and to hold a critter in their hands, it's a really powerful experience, and hopefully it will carry on to when they're older. They can tell their friends about it and go from there. So with that, I would love to answer any questions. Yes. So not necessarily tied. Oh, yes. So he was asking if we have seen any indirect effects or sort of a correlation between the numbers of lake sturgeon and other organisms. So as I mentioned, with the reefs that we installed, lake sturgeon was the primary target for those, but they have definitely benefited other species, and we are seeing other species spawning on them. The big thing being lake whitefish, which hadn't been seen to spawn in the Detroit River in 90 plus years. One of the really cool things is that in addition to catching sturgeon on the set lines, is that they started catching a few northern mad tom in the St. Clair River. And so on the end of each set line, we now put a minnow trap. And so we are now tracking the number of northern mad toms in those minnow traps, and we are actually catching quite a few. And northern mad tom is a threatened species, I believe, in both Michigan and Canada. So that's an example of where we leveraged our work for lake sturgeon to learn more about a species of special concern. Yes.
The reefs that are being built, the sturgeon spawn, are those like off-limit fishing areas that the anglers know about? Or is it someplace anglers go to in order to catch these fish?
Yeah, so the question was, are anglers off limits, or are they using the sturgeon spawning reefs? So these are sited in the mill in pretty deep areas in both the St. Clair River and the Detroit River. They are not officially off limits, but there are not a lot of anglers using them. However, there are some more near shore habitat projects that have gone in around some of the islands in the Detroit River, and they have used a lot of rock to sort of create backwater areas where more wetlands can grow, and anglers are definitely using those rocky spots and catching a lot of fish. Are there any online questions?
So for that, for the middle part of your talk, when you talked about how your catch rates in the Canadian Mart.
Mhm.
So, but you also showed that the catch rates, the angler catch rates, just in general, in Lake St. Clair and Superior are huge.
Yes, yes.
How do those catch rates compare between American waters and then Canadian waters? And are the Canadian waters, providing?
Yes, yes. And so that's one of the things that our anglers always say is, oh, the big perch are in Canada, all the bass are going over into Canada. We do have some folks who do cross the border and they are fishing both waters. They're fishing in Canada or they're fishing in Michigan. We have done some studies, or we've done some telemetry studies with smallmouth bass, and smallmouth bass actually are more homebodied than we thought. Some are crossing the divide. A lot of fish like Anchor Bay, in Anchor Bay. So The catch rates are high on both the American and the Canadian side. Canada only does a creel survey about once every 10 years or so. We've done a creel survey on Lake St. Clair, with the exception of 2 years when we did the Detroit River since 2016. So we don't have like a year-to-year comparison, but angler catch rates are pretty similar between the two sides.
So that's legal?
Yes. Yep.
Why do the island smallmouth bass, they'll go up into here, they'll come up here and they'll come back down, like, what's that all about? They're like, then they come back here.
Yeah, I don't know whether it's a food thing, a temperature thing. So Lake St. Clair, surprisingly, it gets warm, but because of the cold water coming down from Lake Huron through the St. Clair River, it does stay a little bit cooler than Lake Erie. So temperature could play a role there, but yeah, fish are crazy. So we've done a musky telemetry study on Lake St. Clair. And ironically, one of the fish, his tag number was 007. And so James Bond went all the way from Lake St. Clair to Niagara Falls and back in one season. So, yes. Are there any what reasons?
Plans.
Plans. So the U.S. Fish and Wildlife Service, because they have now started stocking fish into the Maumee River, are starting to sample more in the waters of Lake Erie. And they have actually gotten a few recaps from anglers in the commercial fishing of some of those sturgeon that have heads that were put into the Maumee River. So that's only, they've only done that for a few years. So I would expect, as we're putting more fish in and those fish are getting bigger, that once, more years have passed, that there will be a lot more energy. But right now, there's just not a lot of them.
I can say that one of those tagged fish was detected right up here at Split Bay Harbor
Awesome.
off Gibraltar.
Cool.
That came from the Maumee River.
Awesome. Well, thank you so much for giving me your attention on a beautiful Thursday night. And
Thank you so much, Sarah. I'll just add, you did mention the Toledo Zoo sturgeon rearing. So have any of you seen the sturgeon trailer out by the Toledo Aquarium and Zoo? It's really cool. Jeff, you know about it. Yeah, it's really cool. You can, if it's not too crowded, you can go in there and take a tour to see how they rear juvenile lake sturgeon. The importance of it is that they're using Maumee River water in their tanks to imprint on the sturgeon. Can you think of another organism that may imprint on its native streams to migrate? Maybe you eat them. Salmon, yeah. I can hear you starting to eke out the S sound. So sturgeon are kind of like salmon. So that's the idea. Let them grow up in Maumee River water. And they have a festival in October, and you can adopt one of these sturgeon and hold it in your hand and release it into the Maumee. So I encourage you to look that up with the Toledo Zoo and Aquarium, their little sturgeon festival in October. Please go see that. Before you all leave, are there any announcements from professors or anything? If not, what I'd like to do is before you leave, if you enjoyed this seminar and you learned something from it and you think you'll take that information away with you, please take a snap of the QR code. You too, John, online. Thank you for joining us. We appreciate you former Stone Land manager John Hagman's online listening to the talk. Feel free to use that code as well to put your comments into that little survey on this seminar. Next week, we will have Dr. Lindsay Bruckerhoff from OSU. She's a new assistant professor within EEOB. She's going to talk about some fish stream ecology stuff. So please come back and enjoy that. Sarah, I have to get you and your family back to the lighthouse by we'll probably leave around 8:30, 8:35. So if any of you want to chit chat with Sarah after this, I'm sure she would love to talk more fish with you. All right. Thank you, everybody. And that's it.